In this first presentation, we're going to look at the role of the cell membrane in allowing molecules to move in and out of cells. So first of all, we're just going to identify that there is movement of molecules in and out of cells. So we know that for cells to function, there must be exchange of materials. So cellular function only occurs when there is exchange of substances, and this occurs across the cell membrane. So it must occur the cell membrane in plants and animals. Added to this, in plants, it must also be able to occur across the cell wall. So let's revise what's needed across the cell membrane. So we'll just draw just a regular cell membrane, and we'll put in there the nucleus. So that is our cell membrane. We know that there must be movement of materials into and out of cells. So the sort of things that are needed into the cell, we're just going to show that in blue, would be oxygen. And oxygen is required in all cells for respiration. Plants also require carbon dioxide. And that's for photosynthesis. Plants also need a lot of nutrients brought into cells. So these nutrients include glucose for cellular respiration, amino acids for the production of proteins, as well as lipids and water. The sort of things that are needed to get rid of is waste products. I'll show this in red, so our waste products. Include carbon dioxide in all animals as a product of respiration, urea from some cells, uric acid, and excess water. All need to be getting rid of. Last thing is secretions. Okay, we'll show secretions in green. Sort of things that are secreted out of cells include mucus, hormones, and enzymes. So for all this to occur across the cell membrane, then cell membranes must be able to allow things in and out. And they're what we call selectively, so cell membranes are selectively permeable. So they're selectively permeable. Now you'll recall from the experiments we've done in class so far that the experiment highlighted that small substances like iodine, for example, and water were allowed to travel quite easily through the dialysis tubing, which was modelling a cell membrane. Larger substances are also needed. So this presentation will look at how not only the small stuff move through the cell membrane, but also how larger things move through cell membranes. In this next section, we're going to compare the processes of diffusion and osmosis. So on the left-hand side, we can see a diagram representing diffusion. So what we can see on the left-hand side is a far greater concentration of these particles than we can see on the right-hand side. And what will occur is that there will be a shift of these particles from the area of high concentration to the area of low concentration. So that is the process of diffusion, the movement of particles from an area of high concentration to an area of low concentration. Now I'll do this until equilibrium is reached. equilibrium is reached when there is equal amounts of particles on both sides. On the right hand side of the screen we can see osmosis. Now this differs because the big blue ones here, that is our water molecules, and these are our solutes. These are in solution with the water molecules. So what we can see on the left hand side of this line here, and this is a selectively permeable membrane. A selectively permeable membrane is there is far greater concentration of water molecules to the solute molecules compared to the right hand side, where there are far less water molecules and far greater solute concentration. So in this example, in osmosis, there is a movement of water from an area of high concentration of water molecules to an area of low concentration of water molecules. So in both cases we can see there is a movement of particles from a high concentration to a low concentration. So movement from high concentration to low concentration. Now this is also what we call along a concentration gradient. So it occurs along a concentration gradient. So it's shown by a slope going down. It is easier for the particles to move down the slope from area of high concentration to low concentration, and this is what we call along a concentration gradient, compared to against a concentration gradient, which would be upwards, like pushing something uphill. So because it goes along a concentration gradient, there is no energy required. So no energy required. Or it's what we call passive movement. So there is passive movement of these particles from an area of high concentration to low concentration. And there is a passive movement of the water molecules from an area of high water molecule concentration to an area of low water molecule concentration. The definition for diffusion is the movement of any molecule from an area of high concentration to an area of low concentration. Now this occurs until equilibrium is reached. Now it is worth pointing out with this term equilibrium that what equilibrium means is the same concentration particles in both areas. The same concentration of particles. They are still moving. However, there is no net movement. Now, osmosis is a special type of diffusion. It's a special type of diffusion because it relies on the movement of water molecules. So it is the movement of water molecules from an area of high water concentration to an area of low water concentration. Now it also depends on solute concentration. There is a high, so it depends on solute concentration. If there is a high solute concentration, we call that low water molecule concentration, so the movement will be in the direction of the high solute concentration. If there is a low solute concentration, we call that a high water molecule concentration, and the movement will be away. So the two terms kind of mean the same thing. One's talking about the amount of water, one's talking about solute concentration, but they lead to the same concept, that if one is high, the other is actually low. In this section, we're going to look at the current model to explain the movement of substances into and out of cells. So this dot point specifically says, describe the current model of membrane structure and explain how it accounts for the movement of substances into and out of cells. The current model is called the fluid mosaic model. Fluid mosaic model. And this model was first proposed in 1972. Okay, and what this model is in two parts. There are two major components to it with a whole bunch of other chemicals that are involved as well. So the first part of it is what we call the lipid component. So we know that lipids are fats, fatty acids, oils, and those sorts of things. So lipid component. The lipid component consists of a hydrophilic head. So that's our hydrophilic head. So we'll label that. As its name suggests, it has something to do with water, the word hydro. And in fact, the hydrophilic head is what we call attracted to water, or in layman's terms, it is what we call water-loving. Now the tail of it, I'm just going to draw that in white, is hydrophobic. And they repel water, or what we call water hating. That kind of makes sense if we break down that word, hydrophobic. Phobia is a dislike of something, so it hates water, or it repels water. So they're the two parts of our what we call a phospholipid. Let's put that word in. Phospholipid. And that is just one of the components. Now the way that they're arranged, so if we draw in our cell again very quickly, and there's our nucleus. If we just grab one section of that, just that section there, we're going to enlarge that somewhat. So what that consists is what we call a lipid bilayer. So we can see again the water-loving parts to it. So the water-loving heads on the outside, and the water-hating or facing inwards to each other. Okay, so if something is water-hating or doesn't like water, it becomes lipid soluble. So there are hydrophobic tails. And they aren't going to allow water molecules or things that are water soluble to pass through them too easy. On the outside we can see our hydrophilic heads. Okay, so that would be facing the interstitial, the area in between cells, and this would be the cytoplasm. Both areas that would have a high water concentration. So it would make sense that the water-loving, the hydrophilic tails that are attracted to water, would be facing the cytoplasm and the interstitial. However, this in-between section is not going to allow things that are water-soluble to pass through it very, very easily. Okay, so that is our lipid component. The other component is what we call the protein component. So we have our lipid component and our protein component. Okay, and the proteins are scattered throughout. And many of them are suspended within the lipid bilayer. There are a number of different types of proteins. Some of the proteins are what we call integral proteins, and they are embedded within the lipid bilayer. Further to this, some of the integral proteins are actually go all the way through, and they're what we call transmembrane proteins. They're still integral because they're within the lipid bilayer. And finally, there are a number of other proteins which we just call peripheral proteins. So peripheral means towards the surface in this case. Now the proteins are often described as like floating in the lipid bilayer. So they're often described as floating within this bilayer. So in this drawing we can see both the phospholipids with their hydrophilic head and hydrophobic tails, and tails pointing in towards each other. We can also see a number of other things. So that is our phospholipid. We can also see these red things. These red things are cholesterol. And simply they're there for a structural component and play an important part in the structure of it. Okay, we can see a number of other things, and these are our proteins. The first one, the yellow one here, is what we call a transmembrane protein. So this might be for the movement of water, so it might be a channel to allow water to move into and out of the cell. So this is a transmembrane protein. That might be allowing water to move in to the cell. So we can see that it has a passageway there. So this other protein here, which is embedded into the lipid bilayer, is what we call an integral protein. And finally, these ones, this is in the cytoplasm. This is what we call a peripheral protein. And they all have different jobs to do in moving substances into and out of cells. So as I said, the water is able to move through these pores, or what we call transmembrane proteins. And peripheral proteins are responsible for packaging up products of digestion or other products synthesized within the cell and moving them through the lipid bilayer. We also see these integral proteins, which are important role in moving things into and out of the cell. So there are some other components involved within the cell membranes, other components. These include some carbohydrates. Now carbohydrates play a role in cell recognition. Now there are a couple of different types of carbohydrates. So if they are attached to the phospholipid cells, we just draw like this, they would be called glycolipids. If they are attached to a protein molecule, so integral protein molecule just happens to be there, and they're attached to there, that would be called a glycoprotein. Anytime we look at a model, we need to explore the evidence that's used to support that model. So in this section, we're going to have a look at the evidence for the fluid mosaic model. Evidence for fluid mosaic model. So the first bit of evidence is that it allows lipids to pass through it. So it allows lipids to pass through the membrane. Therefore, it must have a lipid basis. If it allowed water molecules or ions to pass through, it wouldn't be a lipid basis. Secondly, the behavior of a membrane. So the behavior. It reseals after puncture. So what this tells us is it must have a fluid structure. Now finally, it was first thought that it was a monolayer. However, this was later disproven when the estimates of how big the cell would be were twice the size of what we estimate a cell size to be. So if it was a monolayer, it had twice the surface area of the cell. So this suggested it was arranged in a bilayer. So we've been exploring how molecules can move across a cell membrane, and we've noted that diffusion and osmosis of molecules can account for some of that movement. However, other molecules don't move so easily. So the movement depends on a couple of things. So movement of molecules across cell membrane permeability is dependent on the following. Now these include the size of the molecule. We already know that small molecules move much easier across a selective permeable membrane than do large. The electrical charge, of those molecules, and whether or not they are lipid soluble. So substances with high membrane permeability include small molecules, and these include our gases, so oxygen, carbon dioxide, move very, very well by diffusion across the cell membrane. And lipid-soluble molecules. So these include, obviously, lipids themselves, urea, and alcohol can also move very, very well across. So alcohol is both is soluble in water and in lipids in varying amounts. Things that have low permeability Low membrane. Water-soluble molecules. And electrical charged molecules. Such as ions. So sodium ions, potassium ions. So these things rely on carrier proteins. So they have low permeability, they rely on carrier proteins. Rely on carrier proteins to move them through. And this is a process what we call facilitated diffusion. So those protein molecules that are within the membrane allow for certain molecules to move through the cell membrane quite much quicker than they could move through without the use of the carrier protein. It's a process called facilitated diffusion, and it is still a form of passive transport. Water molecules are still, although highly charged, are still quite permeable. So water molecules are still quite permeable because they carry through hydrophilic carrier proteins, what we call aquaporons. So the aquaporons are hydrophilic carrier proteins, and these are the carrier proteins that are transmembrane. So they go all the way through the lipid bilayer, and they allow water to travel through the protein rather than through the lipid bilayer, which they can't because they are water, which are not lipid soluble. Now in the last section we looked at this concept of facilitated diffusion. So facilitated diffusion, and that is the use of carrier proteins along a concentration gradient. So they're moving in along a concentration gradient, so from a high concentration to low concentration. And it is still considered passive, so passive diffusion, passive movement. There's no energy input. This leaves the question of what happens when we're trying to accumulate greater amounts of molecules in an area. So say for in the kidneys when it's trying to get rid of urea and more salts. So once the problem exists is once equilibrium is met, there is no more net movement. So the way around this is the idea of what we call active transport. So what we call active transport. So again, if we just show this through a small diagram, and we saw that before with using a slope, but if we this time show the slope in the other direction, and we're trying to move these green molecules from an area of low concentration to an area of high concentration, this wouldn't be possible with passive diffusion. This wouldn't occur naturally. So what's needed to move these guys over here and keep them out of here is the input of energy. So this is active transport and requires energy. There are a number of them, not just the kidneys, but there are a number of situations where active transport is needed. So again, the way this occurs is through carrier proteins. So carrier proteins will channel or will pick up if they're peripheral proteins. So peripheral proteins will engulf the molecules on the inside of the cell or on the outside of the cell and bring it through the other side. So they move molecules against concentration gradient. And that requires energy. So if there are channels, again, the channels draw in more of the molecules. From a low concentration to high concentration. And again, there is a requirement of energy for that to occur. Let's just sum up that idea of active transport. So active transport. So it is the movement of molecules from an area of low concentration to an area of high concentration. And it requires the input of energy. So thank you for looking at this presentation. I hope you've enjoyed it, and I will see you in class.